What are homopolysaccharides? Before we answer that, we must first know what a polysaccharide is. Polysaccharides are carbohydrate chains made of 10 or more monosaccharide residues, joined together by glycosidic bonds. Their primary function is to provide structure and store energy. There are two types of polysaccharides: homopolysaccharides and heteropolysaccharides. This model helps us to illustrate the differences between hetero and homopolysaccharides. As you can see, it is built from similar objects, the objects representing molecules. On the other hand, different molecules help build this chain, making it a heteropolysaccharide. The colors represent different types of molecules. Now that we know what a homopolysaccharide is, we will explore the 4 different types of homopolysaccharides. Cellulose is a homopolysaccharide that consists of linear chains of glucose residues bound by beta-1,4 glycosidic bonds. The cellulose molecule can consist of several hundred to over 10,000 glucose residues. Glucose itself is a simple monosaccharide found in plants and is one of the main products of photosynthesis. Cellulose is strong because its linear chains can form hydrogen bonds with other adjacent chains. This forms rigid sheets of the polymer. Because of its rigidity, many plants use it for structural support, such as trees or celery. The second homopolysaccharide we will talk about is starch. Starch is a homopolysaccharide found in plants and animals that is used to store energy. The two components of starch are amylose and amylopectin. Amylose is the first component of starch and contains chains of glucose residue connected by alpha-1,4 glycosidic bonds. Unlike cellulose, amylose is not linear, but coils into a helix. This is an example of a helix structure. Amylopectin is the second component of starch and, like amylose, it contains glucose chains bound by alpha-1,4 glycosidic bonds. However, unlike amylose, the glucose branches out instead of coiling into helix because of the alpha-1,6 glycosidic bonds that it also contains. The glucose branches can be pruned to release energy at various points simultaneously. Here is a model of a branched amylopectin structure. This model works if we assume that every blue unit represents 25 to 30 glucose molecules, since the frequency of branching is important in distinguishing it from a third type of homopolysaccharide. Starch can be found in all green plants and many of the world's staple foods, such as rice and even unripe bananas.
For relaxing times, make it some sort of time.
The third homopolysaccharide is glycogen. Like amylopectin, it also contains branched monosaccharides linked together by alpha-1,4 glycosidic bonds. The difference between the two is that glycogen, also known as animal starch, is more branched. To give an idea of the frequency of branching that occurs in glycogen compared to amylopectin, the monosaccharides branch every 8 to 12 residues, while amylopectin monosaccharides branch every 25 to 30. Mentioned earlier, this model can also represent the branched glycogen structure. However, for this model to work, we must assume that each blue line represents 8 to 12 residues. This means that the structure branches more and can release energy at much faster speeds. Glycogen is formed in humans in the liver. And can also be found in muscles. When needed, the glycogen is broken down into glucose for more energy. This makes glycogen an extremely important source of surplus energy for our bodies. Lastly, chitin is the fourth homopolysaccharide, and it is composed of N-acetylglucosamine chains that, like the chains of cellulose, form beta 1-4 bonds with each other. Because of the polymer chains in chitin hydrogen bonding with each other, it is very rigid. This is an example of hydrogen bonding between N-acetylglucosamine molecules, and this is an example of hydrogen bonding between N-acetylglucosamine chains. Chitin can be found in the exoskeletons of many insects and arthropods. The Australian wasp is an example of such an insect. Thanks for watching.